This video is a part of the series about RNA-seq data preprocessing using the geneXplain platform. We discuss the mapping step in this video, and you can find links to other parts in the description box. Sequencing machines produce a collection of reads from the sample RNA library. The reads are just sequences of nucleotides that need to be mapped to some reference sequence to get useful information like gene expression levels. The task of assigning the reads to a specific location in the reference is called read alignment or read mapping. This task is not trivial. A read may perfectly match some region of reference. However, this is not always the case. The alignment method should be able to map a read to the best reference position considering possible sequence variants. It can be a single base switch. It can also be a mismatch in part of the sequence with some bases missing in the read or extra bases present compared to the reference. The alignment methods usually have parameters for adjusting how strict is the performed matching. There are several mapping tools available inside the geneXplain platform. They can all be found under RNA-seq analysis from the start page of the platform. Go to the RNA-seq preprocessing category and select Alignment of RNA-seq data. There are several tools like subread, subjunc, HiSet2, and TopHat listed. They can all be used for read mapping tasks and thus have a similar set of core parameters. The selection of a particular method may depend on the necessity to use some additional parameter or to get additional output information. Subread method will be used in this video as an example. Alignment methods accept files in FASTQ or FASTA format. The number of input files depend on the library type used for the sequencing experiment, one for the single end and two for paired end type. We select two input files from the data repository in this example. Let's set the output parameters as well. There are two main outputs, aligned reads and mapping summary. Some alignment tools also produce additional outputs. Subread, for instance, generates a file with detected insertions and deletions. Specify the path and appropriate name for each output. The next important input is the reference genome. The first option is to provide the reference as a file from the data repository. In this case, the mapping tool will index the custom sequence first and then map input reads to the generated index. Another option is to use a pre-built index from the list of common reference genomes available in the platform. In the example, ensemble genome for Homo sapiens is selected for reference. A gene annotation file can be optionally provided to enhance the alignment. More details about the annotation parameter are discussed in the read quantification video. Main parameters are now set. However, more advanced users may want to view the full parameters list. The list changes slightly depending on the tool, and most values are set to defaults suggested by the tool developers. One may need to set a higher thresholds or allow less mismatches for more precise alignment. The minimum and maximum fragment lengths can be adjusted when mapping macroRNA or long sequencing reads. In our example, we use the trimming parameter to discard 15 bases from 5' end because an adapter sequence was detected there during the quality check. A more detailed description of all parameters is available in the info box of the bottom panel. When all the parameters are set, hit the Run button to launch the task. It will take some time for analysis to produce the result. After the completion of the task, it is recommended to check the summary text file and examine the alignment performance. The file saves parameter values and provides alignment statistics like number of mapped and unmapped reads. The actual alignment information comes in the BAM file. BAM stands for Binary Alignment Map, and it is a compressed version of SAM or Sequence Alignment Map file. These file formats are represented by tracks inside the geneXplain platform. Tracks open directly in the genome browser, with each read shown in its assigned genomic position. The sequence reference for a given track can be checked in the info box together with other metadata. Subread also produces a VCF file with found genomic variations. The variant call format file is stored as a track as well, and so can be viewed in Genome browser. Some track files also open as tables for more detailed view. The platform provides annotation tracks which can be useful when working with Genome browser. For instance, ensemble databases include a number of tracks. Let's open Genes track. The reads and annotation can now be visually compared. We see that some regions of genome have more reads assigned to them. Counting how many reads were mapped to specific regions of genome is yet another task. Check the read quantification video to find out how to process BAM file into a count table.